Hello, everyone, and welcome to Cascades Analytics Live. My name is Alexandre Stepanovic, founder of Cascades Analytics, an independent research and advisory platform focused on the neuropharmacology driving the psychedelic and neuroplastogen pipeline. Today's question: how can we measure the signaling bias of the 5-HT2A receptor, the receptor necessary for hallucinations and partially for the therapeutic effect of psychedelics? So my guest today is Dr. Eline Pottie, whose work on the 5-HT2A biased agonism is directly relevant to that question. Hi, Eline, how are you today? Thank you for joining us.
Good evening. Yeah, I'm doing very well. I hope it's the same for you, everyone.
Thank you for joining us. I wanted, to start with a personal question. Maybe it's what drew you to study the 5-HT2A signaling bias specifically, instead of the receptor pharmacology more broadly.
Yes. Actually, it's grown a bit organically, I think. During my studies in pharmaceutical sciences, I was always very much drawn to how can a small molecule, or a rather small molecule, have such an impact on the human body. So I joined the laboratory of toxicology for my PhD research, and there they were interested in having a bioassay which could capture, which could detect the presence of psychedelic substances from a toxicology point of view. And I made that assay. It didn't work for detection because human matrices, they also contain serotonin, which is an issue in the detection. But anyway, by then I got much intrigued by psychedelics because it's undeniable that they have very profound effects on the human mind and body as well. We were interested in knowing why these compounds do what they do, and if there's differences between different chemical classes in what they do. So we started with the beta-arrestin assay that we had set up at that point already, and then we complemented it with a G protein assay that we still needed to set up, and then it just started from there. And it went all over the place, I guess.
And could you tell us about the bioassays you developed and how they are different from the existing bioassays? Is there any bioassay of reference to study GQ versus beta-arrestin?
Yeah, sure. I have a few slides, so maybe it's easiest if I have some visual. How do you say it? So if you look at the serotonin 2A receptors, there's actually quite a lot of signaling pathways or interacting proteins that have been identified. This isn't even a full list. There's even a whole lot more proteins and effects that this receptor can result in, activation of the receptor can result in. So what's typically looked at is GQ signaling and beta-arrestin 2 recruitment, also a bit at beta-arrestin 1, but to a lesser extent, I have the impression. So we already had a beta-arrestin 2 recruitment assay, and this is something that happens very fast or very close to the receptor still, because it has a direct interaction with the receptor. So we thought that was an advantage because, as you will see later, our receptor is tagged. It has a protein that is fused to it, and the beta-arrestin has a protein that is fused to it. So if you measure a signal, you're 100% sure that you're measuring the interaction between the receptor and the protein, and not the response of another receptor which is expressed in the cells that you are looking at. So that's something that we kept in the back of our minds when we looked at the G protein signaling pathway as well. So there's quite a lot of assays that are frequently used here in that pathway. I think the calcium assay is probably the most frequently used. The reason that we did not choose the calcium assay is because it is difficult, or it has some specific implications if you want to measure slowly binding agonists, because it's a very fast signaling event, and it's a bit more difficult to capture slowly binding agonists. And for example, if you think of LSD, it is known that it is very slowly binding to the receptor, so you would probably underestimate the effects. That's something that you need to consider. And we didn't want to do.
Sorry, Eline, when you say slowly binding, you mean the retention at the receptor, or the
Yes.
retention time, or?
Yeah, it stays at the receptor for a very long time.
Right.
There's also, for example, IP3. This is also very frequently monitored signaling event. Actually, we have tried doing an assay, and actually it's very convenient measuring it. But also here, we took a bit into account that there may be other G protein coupled receptors that activate the GQ signaling cascade, and you can't really be sure that the IP3 that you're measuring is actually coming from your serotonin 2A receptor. And we, for the beginning, really wanted to stick with the serotonin 2A receptor. So that's a few considerations why we didn't select the ones that are very frequently used.
Are you looking in neurons or in cells, and what type of cells? And do these cells have the same proteome than the neurons in the prefrontal cortex, for example?
So we're working with HEK cells, human embryonic kidney cells. So there's an immortalized cell line. This is obviously not the same as measuring in neurons, for example. So the receptor expression pattern is much different. I think I haven't really looked very much into specific types of neurons that you can look at, because also it's quite a dedicated culturing technique. So as we started from the toxicology point of view, we started just with cells which are easy to work with, and then HEK cells definitely made it to the top of the list.
Right, and it's transfected 5-HT2A.
We do transient transfection. There's also other, there's some serotonin receptors which are endogenously expressed in HEK cells, such as the serotonin 7 receptor, but it doesn't signal through GQ mostly. So that's not too much of an issue for this purpose. So here we took into account the fact that if you start close to the receptor, the further along you go, the more the signal gets amplified, which also complicates your interpretation. And then what I said about the signal interferences, so there can be interference from other receptors. And specifically, as it's in HEK cells, we don't have the receptor expression pattern that would be physiologically relevant. So it's best not to have that interference at all. So if you move further downstream, the complexity of the interpretation actually increases, which is why we wanted to stay upstream. And we work with a mini G protein, which is an engineered G-alpha-Q subunit. We haven't engineered it. We have just fused part of a luciferase enzyme. So both of our receptors actually work via of our receptor systems, of our activation assays, work via a very similar principle. So they work with the nanobit technique, where a small luciferase enzyme is split into two parts. When the two parts are separate, there's no luminescence. If the two parts are together, then you can measure a luminescent signal. So in our case, we've used one part of that luciferase to the receptor. the other part to either the mini-G protein or beta-arrestin. And then if you add the psychedelic or the other serotonin also obviously work, whatever serotonin to A receptor agonist you want to add, the intracellular protein gets recruited, and you can measure luminescence. And from this, we can then...
One quick question, sorry. Is the luminescence proportional to the amount
Yes.
of... Yeah.
Yeah. So what we end up with eventually is a concentration response curve.
Mm-hmm.
So here, each of those points is actually derived from the area under the curve of the luminescence profile. So we measure luminescence continuously for 2 hours.
Mm-hmm.
So each well of a 96 well plate is measured every 2 minutes. So we can generate either a curve which shows increase in luminescence or a flat curve, which is when you don't add anything. And then we calculate the area under that curve, and that then gets plotted in such a sigmoidal curve. From this, you can derive the efficacy, or the Emax, which says something about how strong your effect can be. For example, in this case, the compound for which the curve is depicted in black is more efficacious than the compound for which the curve is depicted in gray, because it's lower, the gray one. And you can also say something about the potency, or the EC50 value. This is the concentration that is required to get half of your maximal effect. Also here, the black, or the compound for which the curve is depicted in black, is more potent because it needs a lower concentration of the compound to get half of its maximal effects. And this is more or less what we start from with our assays.
Great. Are the beta-arrestin bioluminescence assay and the GQ protein or mini GQ protein bioluminescence assay mirroring each other?
What do you mean by...
It's one or the other after...
Yeah, it's one or the other. So we have hexels which express the receptor and the mini-G protein, and we have other hexels completely separate, which express the serotonin receptor to A receptor and beta-arrestin 2. They're separate from each other. The protocols are very, very similar. So we transfect the same quantities of receptor. We use the same buffer. We use the same amount of substrate and so on. But they are in separate cells.
I see. Great.
Yes. So what we then do to estimate biased agonism, I don't know if you want to go further on this, or if you...
Oh, yes, please.
Yeah. Okay, so we then have EC50 and Emax values in both of these assays. And then we use a formula, which is depicted here, but it's a bit small. So we calculate the relative activity of each compound in each pathway. Pathway or signaling event or assay, just to be entirely correct. And this RAI, it's the ratio of Emax to EC50 of your compound in which you're interested, relative to the Emax to EC50 ratio of your reference agonist at that point. So you can actually generate RAI values for basically every compound that you're measuring, as long as you have a reference. And then we combine those two RAI values into a bias factor, beta. So this bias factor, it says something about how strong your preference is relative to a reference agonist, because you took along the EC50 and the Emax values of your reference agonist as well. So in this case, LSD was our reference agonist, because you're taking the ratio of your EC50 and Emax. The bias factor of your reference compound is 0. and everything else is relative to that.
Why did you take LSD as a reference and not serotonin, for example?
Yes, that's a very interesting question, and we actually get it every time. So now we take serotonin as a reference because it does make more sense. At that point, we were still too much into only looking at psychedelics. And when you're only looking at psychedelics, LSD sort of makes sense because it's such a well-known compound. But basically, you can calculate bias relative to everything. There's 3 types of bias you can look at. So there's physiology bias if you take your endogenous agonist as a reference compound, which you would calculate if you would measure relative to serotonin. There's true bias, so to say, but that then you have to have a compound for which it is known that it does equally activate the 2 pathways, and it's very difficult to prove that it really does and that it's not an artifact of your assays, for example. I don't know if any such agonist exists for the serotonin 2A receptor, to be honest. And then you can just, a third option is to have benchmark bias, it's called, and then you can just calculate it relative to every compound on which you're interested. And this is what we did here. We had a look at serotonin, at LSD, I'm sorry, as a relevant compound relative to which we want to know whether, towards which pathway it's, or assay it's more biased.
But isn't LSD tricky because it's psychedelic, hallucinogenic, but also has a beta-arrestin component, I reckon?
Yes. We realized that afterwards. So yeah, you're here very much dependent on your reference agonist. Definitely.
And so here we have the range under LSD reference of, let's say the beta oscillates between 0.34, sorry.
Yeah, 0.2, I think, and 0.8-ish.
Yeah. Okay, yeah, under 1 always.
Yeah.
So it's quite a big range.
Yes.
And most of them are under 0.5.
Yeah, I think so.
Yeah, 3/4. So they are mostly beta-arrestin here?
No, in this case, yeah, they are beta-arrestin. Yes, you're correct.
So, but that's compared to LSD. So that's interesting because LSD is already beta-arrestin.
Yes. So that's something that I think is very difficult to compare with literature because we're capturing the bias factor into just one value, whereas it takes into account the EC50 and the Emax of your reference, and the EC50 and the Emax of your test compound, and that for both assays. So you're actually compiling so many data, which can be so variable, and also if the ratios change, the value can change a lot or can just change a little. So a lot is happening actually in there, and we're just trying to compile it all into one bias factor. It does feel a bit uncomfortable at times to reduce the data to just one value.
Sure. All these compounds, do they all have roughly the same retention time at the receptor?
We don't know. So
these are all NPS?
Yes. So I don't know if all of them have been captured on the drug market, so to say. Yeah, there's also serotonin, for example. There's NBMD and MBF. they're a bit less active than, for example, the N-bomies and the N-bos. Then there's, for example, 2CD. It's been known for quite a long time already. There's the compound which is called here 1D. It was a test compound that a medicinal chemist made and wanted to have tested, so that's also included here. Yeah, so there's a bit of everything.
Okay.
It started with the MPS, and then people started contacting us, like, we want to know what the compounds do in our assay. Do you want to test it? And then we went from there.
Great.
Yes, I think I also have, I also have it here. So it's very much dependent on your the bias factor. It's very much dependent on your reference agonist that you are using. So in this case here, for example, this is relative to LSD, which is what is called benchmark bias. As I mentioned before, if you would, for example, take serotonin in the two assays that we are using as your reference compounds, then you see that the shape that the curve follows, it stays more or less the same. It's not actually a curve, but like the trend in bias factors, it stays the same, but it starts at a different value, and then you have serotonin, which is 0 at this point. So then most of the compounds lie in the mini G area.
Versus serotonin, you mean? Interesting. Okay. Yeah, the curve on the left seems a little bit more compressed, but it's the same.
Yeah, it's the same.
Trend.
It just has a different value for 0, so to say. It just shifted a bit. So given that it's quite difficult to look at the bias factor as just one value, we also started looking at other ways of looking at the bias, where we also use, for example, bias plots. And each of those bias plots in black shows what a curve, relative curve of mini G activation, or it's not actually activation, mini G recruitment versus beta-arrestin recruitment looks like for the reference agonist. And then you have in red the compound, the curve for that compound, where it also is mini G activation relative to beta-arrestin 2 recruitment, mini GQ recruitment, I'm sorry. It's the mini G, it can't get activated, so it's not correct to say mini G activation. So it's recruitment of mini G or recruitment of beta-arrestin. So also here for the red curve then shows what your test compound does. And for some compounds, it's quite clear. So for example, on top, there's 25N NBF relative to LSD. It's completely in the bias plot is completely in the part below that of the reference agonist, which shows that at concentrations where you still have low activation recruitment of mini GQ, you already have high recruitment of beta-arrestin, which means that it's relatively beta-arrestin 2 preferring relative to your reference agonist. Then for 2CP, it's the other way around. So the curve is on top towards the mini GQ recruitment assay. So at concentrations where there's still low recruitment of beta-arrestin, you already get high recruitment of mini G. And then there are some compounds for which it's a bit more difficult, such as DOT on the right. It starts at one side of the...
It's dose dependent, The bias becomes dose dependent.
Yes. Wow. So that's also one of the reasons that it's very difficult to capture the whole spectrum of activation in the different assays into one factor. If you would only look at the bias factor, you would never have captured that it shifts at a certain point, that it starts at low concentrations at the beta-arrestin site and then at higher concentrations at the mini-G sites.
Is that happening a lot? That in,
Yes.
in many compounds you've screened
Yes.
this dual response?
Yes.
Wow.
Actually, if you look very, very closely to 2CP, actually, I think it's also not the perfect example.
Oh yeah, slightly.
Yeah, actually, it also starts a little bit towards the beta-arrestin site. This can also have to do with the fact that maybe at lower concentrations, the beta-arrestin assay is just a bit more sensitive than the mini-G assay. But it's difficult to tell.
It could be like an intrinsic
Yes.
intrinsic response of the
Yeah.
5-HT2A that once it's bound to its ligand, it's gonna be internalized no matter what. So there has to be some basal arrestin activity going on.
Yeah,
for
can be.
all ligands. Yeah.
Yes.
Great.
Yeah. And then we, as a third measure, we also had a look at just the relative rank order. We ranked the compounds either based on EC50 or on Emax. In this case, it's Emax in both assays. But there's a few, it's interesting to see because from time to time it confirms what you wanted to see. And also it's interesting to see it about, if you're only interested in one measure, only in potency or in efficacy, it's interesting to look at it. But you have to keep in mind that it's only looking at one measure. And also it's independent of a reference agonist. So it depends a bit on which set of compounds you pulled together, whether or not a compound will be relatively more beta-arrestin or relatively more mini-G preferring. And something else that you can consider here is for our assay, it's not very relevant. But if you, for example, take an assay where there's a lot of signal amplification, then it can be that all your values for Emax are around 100%. And that's a certain compound, it's 99.3%, and the other one is 99.5%, which statistically is exactly the same. But then you rank them because you want to rank them and you're using this measure. And then it actually looks like you have huge differences between the compounds, whereas all Emax values may be exactly the same statistically. That's a risk of those assays as well.
Yes. So we're going to talk about psychedelic effects on that graph. Is the psychedelic effect going correlating with those dots? Meaning, the further you go on the median, the more psychedelic effect you get?
We haven't really correlated it here. I know at some point we tried to correlate doses that we found online for the compounds which have been reported as MPS, and we tried to correlate that with the EC50 values in our beta-arrestin assay, and there we saw a correlation. However, it only takes into account those compounds which actually show psychedelic activity, because for other compounds there's no dose to be found because it's not active. So it's quite difficult actually to look into that.
Here you're looking basically at response that are very proximal to the 5-HT2A receptor activation. What if you were looking at more distal activity? Does that graph holds on? Is it still roughly the same if you look at more distal activation? Like, I don't know, you mentioned calcium, but you don't do calcium assays, but IP3
I've
or
done it.
DAG?
I've done it, but I've also set up another assay, which because I made it, I preferentially use it. So it's an interaction assay between GQ and PLC beta. So it works also via the nanobit technique. Actually, we see that if you look at the trends of compounds, it's a different set of compounds entirely. We compared with the mini-G assay, which is most upstream, then the interaction assay between GQ and PLC beta, then the IP1 assay, which is a commercially available IP3 accumulation assay, and then a calcium mobilization assay. And we saw that the three most upstream assays showed very similar trends. Like if you would generate a structure-activity relationship from that, your results would be much similar. And also in terms of correlation, we found correlations. With calcium, it's more difficult because the three upstream assays we all measured for 2 hours, and the calcium assay we only measured for a shorter period of time, where then we had underestimation of the activity of certain compounds, mostly LSD, but then also lizarite, for example. So it didn't correlate very well. We didn't look into bias yet. At that point, we were just actually establishing the assay and looking at whether or not it correlated with other G protein measures, but we haven't used it for bias yet. So that's something that will have to be confirmed.
Super interesting. What about the therapeutic, or let's say the plasticity? Any of these compound, did you see with this graph a correlation with some of the potential, dendritic spines and density or new spine formation, a correlation there?
Yeah, that's a very interesting question, but again, there's quite a bit of difficulties. So I think the first one being the fact that I don't really think there's a consensus yet on which pathway is linked to which effects. So that's something difficult, but also it's very, we're only looking here at the receptor activation,
right?
Which is obviously, it's very important, but there's a lot of other things that happen between taking the compounds and it's finally reaching the receptor. So that's also something that needs to be considered. So there's quite a barrier in translating the in vitro effects to in vivo effects. And also, for this set of compounds, I don't really think that a lot has been reported on the neuroplastogenicity for example, the NBFs or NBMDs. So it's still difficult on many levels. It would be very interesting to do at some point. But at this point, we haven't looked into it yet. There's so much to do.
I know, yes. And looking at the current neuroplastogens in development, what are your thoughts on their low GQ activity and partial agonism? Have you looked into that? Which one do you think your data actually support, and which one do you think are still unresolved, and thinking about Delix and the Gilgamesh compound and stuff like that?
I think it's very intriguing to see how the field evolves. That's for sure. The past, I really haven't tested any of the typical neuroplastogens. What we did see before was, for example, that 2-bromo-LSD and lizarite, they indeed have lower efficacy at the serotonin 2A receptor, also in our assays, consistently.
So it's not partial agonism, it's lower efficacy.
Yeah, so partial agonism is lower efficacy.
Oh, just in general? Okay.
I think that's what I understand under the term partial agonism, so that you don't reach 100% of receptor activation.
Yes, but you could reach 10% of both pathways. That would be low efficacy, but not really a partial agonism.
Okay, that way. It does, in our assays, activate the receptor to like 50% of serotonin. So I don't know if you would consider that a partial agonist or not.
Right. Yeah. Okay. It's just what you just said about those neuroplastogen. There was an idea that they were probably they had a beta factor. From my understanding, you'd said they'd have a beta factor favoring beta-arrestin versus GQ, but with a low efficacy. So that means that they don't do anything, basically. They activate very little. They do activate a little bit of beta-arrestin, but the beta factor in itself doesn't predict the efficacy overall, like the floor the absolute floor efficacy, how much you're gonna get downstream activated. You see what I mean?
Not really, actually.
Let me try to clarify. So if you activate 5-HT2A and you claim that you don't have, it's a neuroplastogen, you avoid the psychedelic effect because you claim that you have a more beta-arrestin biased agonist.
Yes.
But overall, you have a very low efficacy. So even if you favor beta-arrestin, you don't get much activation downstream.
Yes. Okay.
And that, I think, can be a problem with in the field of neuroplastogen. They're claiming they're having beta-arrestin partial agonism bias, but it doesn't prove there is actually efficacy.
Okay. Yeah, I see what you mean, but I don't really know about what the compounds do in our assays.
Right, that's true. Yeah.
If we only look at one assay, I know that they have efficacy of 50%, for example, in the beta-arrestin assay or in the mini-G assay as well. And that their potencies are comparable to other compounds. So, but I don't know which extent of receptor activation you require to get a neuroplastogenic effect or to get a psychedelic effect.
Yeah, exactly. So it's the idea that the beta factor is going to tell you what pathways does the ligand prefers, but doesn't mean there's a efficacy. It doesn't mean it's potent. Just that's the I think that's kind of the mirage we've been having lately, is that it's more a beta-arrestin. Yeah, but if it's biased toward beta-arrestin, doesn't mean it's potent at all.
Yeah.
So
okay.
It's more qualitative. The beta factor is more qualitative than quantitative.
Yes. Yeah. Obviously, that's also a shortcoming to the bias factor. That's you don't see the EC50 and the Emax of your individual compounds anymore. So if you get low activity in both assays, but you get 8% versus 10%, then
exactly.
Yeah. Okay.
It kind of suppresses that, and
yes.
And it's just a reminder that we're losing that important component, which is
Yes.
efficacy,
Yeah.
absolute efficacy, let's say.
Yes.
Yeah.
Totally agree.
Well, is there anything you want to add? Is there any new bioassay you're developing right now? Are you moving to neurons, or is it still too difficult?
At the moment, I'm still working on a few projects with the PLC beta assay, the GQ PLC beta assay. It's very interesting. I know we're talking about the 2A receptor, but the fact that there's other serotonin receptors which are also GQ coupled makes it relatively easy. It's always dangerous to say that something in research will be easy because it ends up being very difficult. But yeah, you could use the same assay for the serotonin 2B and the serotonin 2C receptor. So I think we'll look into that a bit. And I'm not really planning very much upfront because my funding only takes me 6 more months. And then we'll see if I can secure more funding and more research.
Well, I hope you will. I hope you will. Thank you so much, Eline, for coming with us to this.
Thank you for inviting me.
Yes, of course. Stay with me for a second, and I'll just thank you all for tuning in. And if you have any question, feel free to DM me or email me, alex@cascades-analytics.com. Thank you very much, and see you next week for another episode of Cascade Analytics Live. Thank you so much.